Germs are all around us, both in our environment and in our bodies. When a person is susceptible and they encounter a harmful organism, it can lead to disease or even death. The body has many ways of defending itself against pathogens like skin, mucus, and cilia, and all work as physical barriers to prevent pathogens from entering the body in the first place. When a pathogen does infect the body, our body's defenses, called the immune system, are triggered and the pathogen is attacked and destroyed. Each pathogen is made up of several subparts, usually unique to that specific pathogen and the disease it causes. The subpart of a pathogen that causes the formation of antibodies is called an antigen. You can consider antibodies as the soldiers in your body's defense system. Each antibody, or soldier, in our system is trained to recognize one specific antigen. We have thousands of different antibodies in our bodies. When the human body is exposed to an antigen for the first time, it takes time for the immune system to respond and produce antibodies specific to that antigen. In the meantime, the person is susceptible to becoming ill. Once the antigen-specific antibodies are produced, they work with the rest of the immune system to destroy the pathogen and stop the disease. Once the body produces antibodies in its primary response to an antigen, it also creates antibody-producing memory cells, which remain alive even after the pathogen is defeated by the antibodies. If the body is exposed to the same pathogen more than once, the antibody response is much faster and more effective than the first time around because the memory cells are at the ready to pump out antibodies against that antigen. Vaccines contain weakened or inactive parts of a particular organism or antigen that triggers an immune response within the body. Newer vaccines contain the blueprint for producing antigens rather than the antigen itself. Regardless of whether the vaccine is made up of the antigen itself or the blueprint, so that the body will produce the antigen, this weakened version will not cause the disease in the person receiving the vaccine, but it will prompt their immune system to respond much as it would have on its first reaction to the actual pathogen. Some vaccines require multiple doses, given weeks or months apart. This is sometimes needed to allow for the production of long-lived antibodies and development of memory cells. In this way, the body is trained to fight the specific disease-causing organism, building up memory of the pathogen so as to rapidly fight it if and when exposed in the future. In the case of COVID-19, the antigen is typically the characteristic spike protein found on the surface of the virus, which it normally uses to help it invade human cells. There are 4 categories of vaccines in clinical trials: whole virus, protein subunit, viral vector, and nucleic acids. Some of them try to smuggle the antigen into the body, others use the body's own cells to make the viral antigen. Many conventional vaccines use whole viruses to trigger an immune response. There are 2 main approaches. Live attenuated vaccines use a weakened form of the virus that can still replicate without causing illness. Inactivated vaccines use viruses whose genetic material has been destroyed so they cannot replicate but can still trigger an immune response. Subunit vaccines use pieces of the pathogen, often fragments of protein, to trigger an immune response. Doing so minimizes the risk of side effects, but it also means the immune response may be weaker. This is why they often require adjuvants to help boost the immune response. Nucleic acid vaccines use genetic material, either RNA or DNA, to provide cells with the instructions to make the antigen. In the case of COVID-19, this is usually the viral spike protein. Once this genetic material gets into human cells, it uses our cells' protein factories to make the antigen that will trigger an immune response. The advantages of such vaccines are that they are easy to make and cheap. Since the antigen is produced inside our own cells and in large quantities, the immune reaction should be strong. Viral vector vaccines also work by giving cells genetic instructions to produce antigens. But they differ from nucleic acid vaccines in that they use a harmless virus, different from the one the vaccine is targeting, to deliver these instructions into the cell.